Welcome to Chapter 2. We've gone through Chapter 1, so now we're going to go and do Chapter 2, which is, we're talking chemistry. I know a lot of you have probably not had too much chemistry, so I want to go through this with you to help you out. And start of chemistry, we're looking at obviously the study of composition and properties of matter and how they interact. Matter is anything that takes up space and has mass. It's a little bit different than weight, but anything that has oomph in it, if you would. And when you put a gravitational pull on that mass, it has weight. So when we're talking about chemistry, we're looking at these properties of these elements and what they do. And biochemistry is really where we look at the study of these physiological processes, putting chemistry into biology, and we're looking at these diseases. Okay, so the first thing we have to look at is elements. And some of you have probably seen a periodic table of elements, and these are the naturally occurring types of matter that we have. They have specific names, but we have 92 of them naturally occurring, and we have some that have been artificially made. When we put them together, if we put one element with another element, we have a compound. Now, these elements are made up of atoms, and each element has a different number of particles in that atom, and we're going to be going over that. But we have different elements, and we'll talk about that are required for the body. We have these elements that we need constantly. The main ones, what we call Ch, carbon, hydrogen, oxygen, nitrogen, and then we also have to have sulfur and phosphorus. So if you see these initials, that is the chemical symbol and tells us what that is. Then we have trace elements that we require in small amounts, such as iron and iodine. And then we have ultra trace elements that we only need tiny amount of. All right, so when we look at this, our smallest particle that we have of an element is called an atom. And that should be an element. It has certain pieces to it. It has a nucleus. The nucleus has protons in it and neutrons in it. And then around that atom, it has electrons. Okay? So the nucleus is made up of protons. And we'll take a look at this, but it's a large particle. It has a positive charge. And then in that nucleus, we have sometimes the same number of neutrons, sometimes not. But it's, again, a large particle, but it's uncharged, so it doesn't change the charge of that nucleus. Around the nucleus, we have electrons. They are small, and they have a negative charge. They're in constant motion. Okay, so those are some of the terms. We'll get into what an ion is, but basically what's going on is that normally we have an atom and it's neutrally charged. If we change the charge on it by losing or gaining electrons, and we'll show you how to do that, then it is called an ion. Okay. All right, so these are some of the major elements that we have in the human body, and we talked about our Ch. We have to have calcium and phosphorus and some of these other things that you'll be introduced to throughout anatomy and physiology, and that makes up most of the body. And then we have these trace elements that are important for certain processes that we're going to be doing. So here's our structure of our atom, and this is lithium. This is on our periodic table, would be element number 3, and we know that because there are 3 protons. The number of protons is the element number. Okay, so, or what we call the atomic number. And so this has 3, and it happens to have 4 neutrons, but that's okay because that doesn't have a charge. But around the nucleus are 3 electrons. So you have 3 positive charges, 3 negative charges. So you have a neutral atom. Okay? So the atomic number, the number of protons in the nucleus, and that determines the name of that element. So if there is 1, it is hydrogen. If there is 2, it is helium, and so on. So each element has a unique atomic number, and that identifies that element. The mass number is the number of protons plus the number of neutrons in an atom. Okay, so we looked at that lithium, and it had 3 protons and it had 4 neutrons, so the mass number would be around 7. All right, so for isotopes that we talked about, isotopes have the same atomic number because they have the same number of protons. What changes in an isotope are the number of neutrons, okay? And so you may have, again, 3 neutrons, 4 neutrons, 5 neutrons, whatever, but you have the same number of protons and electrons. It's not an ion, it's an isotope. And so ions are where we lose electrons or gain electrons. An isotope is where we change the number of neutrons. So if we look at oxygen, oxygen forms these 3 isotopes. And so we write oxygen, which is O, and this tells us how many different neutrons are in there. Okay? All right. Radioactive isotopes are not stable. They will release energy in the form of radiation until they lose those particles until they get to a stable form. All right. So sometimes you'll hear the term atomic weight, and that is just basically where you average all the mass numbers that a particular element can have, and you take that average, and that is their atomic weight. All right. So those are the elements. Well, most of what we use, we put these together in different things. So we make molecules and we make compounds, and basically a molecule is where we take 2 or more atoms and combine them. They can be the same element, they can be different elements. So when we have a molecule, we have 2 or more atoms that chemically combine. For example, that we have down here a molecule of hydrogen, H2. That's the usual form of hydrogen. O2, oxygen, is the usual form of oxygen. So those are molecules. So that is when it forms 2 or more atoms and they chemically combine. Now, a compound forms when you take 2 or more atoms of different elements and combine them. For example, glucose. We've got some carbon in there, we've got some hydrogen in there, we've got oxygen. Now, how do we know what's in this particular compound or what's in this molecule? We have what's called a molecular formula, and that tells us the elements that are there and how many of each element is present. So if I look at this is glucose. This tells me that I have 6 carbon atoms, 12 hydrogen, and 6 oxygen. Water, 2 hydrogen, 1 oxygen. And there's a reason for the way these combine, and we'll take a look at that a little bit later on. So you can see how these combine. Here's our H2, there's our O2 that we talked about, and here's water, okay, when it combines. So there are different ways of forming chemical bonds, and basically a chemical bond forms when these atoms combine. What is important when you form a chemical bond are the electrons, because that's what you're really working with. You don't change the nucleus of the atom, you move the electrons around. All right, so the electrons occupy regions around that nucleus in what we call shells. And these shells are in different levels, and I'll show you in a second. But the first shell can hold 2 electrons, second shell 8, third shell 8. And that's all you need to know for this course. All right, the lower shells, the inner shell gets filled first. So the first shell that has 2 electrons gets filled first, and then you move out. The electrons in the outer shell determine how that atom is going to react. Is it not going to react? Is it going to be inert because that shell is full? Or is it an atom that wants to give electrons up because it's got this lonely little electron sitting out there? Or do we want to work with something? You want to grab one because I got 7 electrons in my outer shell and I want another one, so it's full. So that really determines how these atoms react. So if we look at hydrogen, it has that first shell. It has 1 lowly little electron. It is not happy. It wants that shell full. So hydrogen is going to be one of these that's going to want to tie to a lot of things so that it can have a full shell. Helium is full. It's perfectly happy. It's not going to react too much. And we call these usually these inert gases, because they have these full shells and they don't want to react with anything. Lithium, same thing. Here we're going, this is element 3. We have the inner shell full, and the second shell has this little electron out there. I want to combine with something. Okay? So if we look at sodium, here we have an example of a full first shell. We have 8 electrons in the second shell, so that's full. And then we have this lowly little electron on the outside. So it wants to interact because of this lonely little guy here. Okay, if that makes sense. So we have several different kinds of bonds that we can form because of this chemical structure. The first is called an ionic bond. And this is where you are working with an ion. And an ion, again, electrically charged atom, where you have an electrical charge. And what happens is that atom is going to lose or gain the electrons to become stable. So if you've got 1 or 2 electrons sitting out there in that outer shell, in that shell number 2, or with our hydrogen shell number, that lower shell, it's going to want to lose it. Whereas those that have almost full shells, they want to gain them. And that's what happens. So you have cations and anions. A cation is a positively charged ion that forms when it loses an electron. So if you donate your electron, we'll take a look at this in a second. If you donate your electron, you've lost a negative charge. That makes that ion positively charged. That atom is now positively charged, so we call it an ion. The reverse is if we gain an electron, if we fill that shell, then we've added a negative charge, and so that atom becomes a charged ion with a negative charge on it. We call that an anion. The ionic bonds form when we have that kind of situation. You have a very strong bond that forms when you've got these opposite charges attracting and you want to get it. So let's take a look at this. This is sodium, this is chlorine, okay? So you can see we have the sodium atom, we've got this 1 little lonely electron on the outside, and here's chlorine. It is missing 1 electron to fill its outer shell. This is a perfect match. I can take this one and I can move this over here. You have an attraction, And basically what happens is the sodium loses its outer electron, becomes positively charged, and this is how we note it. It is now a sodium ion versus a sodium atom, and we note that it has this positive charge. For chlorine, it gains that electron, and we change from a chlorine atom to a chloride ion, and we show the negative charge. So that's what an ionic bond does, and it's usually with these elements like the sodium, like potassium, that sort of thing, where you want to get rid or gain one or two electrons. All right, so the attraction is between these opposite charges, and that forms a really nice bond. And when you do that, you take sodium, you mix it with chloride, you get salt, the salt that we usually use, and so you can see that. So that's how an ionic bond is formed. The other bond that we use is called a covalent bond. An ionic bond, they don't share well. They are literally grabbing these electrons and moving them over, either losing it or gaining it. Covalent bonds, they're friendly, they share. So these are chemical bonds where the atoms actually share the electrons between the two. So you have like two atoms of hydrogen, like we talked about. They can combine and form a hydrogen molecule, and both these atoms will be stable, and they're going to share this. The same thing happens with water. So let's look at our hydrogen. Here's our hydrogen atom, another one. We've got this lowly little electron out here, and the poor little lonely electrons will be shared between the two, and we have now two hydrogen atoms with basically full shells. So that now they're happy, if you will. Same thing with water. Okay, so here we have oxygen, and it can share this. We talked about usually oxygen goes with two. Well, oxygen will take it needs two electrons to fill its outer shell. So you can take two oxygen molecules, let them share together in a covalent bond, and they're happy. You can actually now bring in two hydrogen atoms and make water molecules, because again, oxygen wants to fill it, and hydrogen with these two can fill it. Okay. All right, when we have a covalent bond, we can have what's called nonpolar covalent bonds and polar covalent bonds. Nonpolar means they are shared equally. These guys are happy, they're very friendly, they share equally, and they basically they say that these atoms have the same electronegativity, their pull, how much they want to gain or lose these electrons. So the atoms of same elements that have the same number of protons, they pull these electrons equally, and so you don't end up with, more positive charge on one side versus more negative charge on another. The polar covalent bonds don't share equally. They are sharing, but they're not sharing equally. So one tends to grab a little more than the other. So you have an unequal charge distribution, and usually the atoms with the larger number of protons have the larger pull. Good example is water. All right, so this would be a polar molecule. This is still a covalent bond because they're sharing, but you have slightly positive ends because of the hydrogen and a slightly negative end because the sharing is occurring here. All right, so the electrons are basically being pulled in one direction or another. Hydrogen bonds are another bond that forms. This is a very weak bond that happens. Now you're going to put some water molecules together, and this is where you put one polar molecule and another polar molecule together, and the slightly positive and negative ends interact. So here we have our lovely little water molecules, and you can see that the slightly positive side is attracted to the slightly negative side, and you get these are called hydrogen bonds, because it's really hydrogen that's doing this, that bonds us together. This becomes important with water. This gives us the tensile strength of water. It's, really hard to break. All right, so other things we're going to talk about: electrolytes, acids, bases, and salts. Electrolytes are substances that when you put them in water, break into their ions. So when you put salt into water, it breaks into sodium ions and chloride ions, and you can conduct electricity with it. So we call it an electrolyte. You'll see this in reports. The doctor will order electrolytes. Well, what he's looking for is sodium and chloride, and wanting to know those levels. An acid and base. We'll look at these tables next, but acids are electrolytes that release hydrogen ions, and the higher the hydrogen ion concentration, that's what we'll talk about. That is really what the pH is looking at. We're looking at hydrogen ion concentration. Bases are substances that release ions that can combine with these. All right, and a salt is formed when you take these electrolytes and you get this interaction between an acid and a base, hydrochloric acid, sodium hydroxide. You get salt and water. So when we talk about acids and bases, what we're looking at, as I mentioned before, is we're measuring the hydrogen ion concentration in a solution. Okay, so when you put this ionically bonded substance into water, those ions are attracted to the slightly charged ends of those polar water molecules that we just looked at. And so they disassociate and they get surrounded by these water molecules, and that's what we call an electrolyte right here, and that can conduct a current. We can measure this. This is when you have an EKG done of your heart. We're looking at the electrical conduction through the heart, and it is because we have this dissociation. This is when you put it, mainly water. You put sodium chloride in water, you're going to get an electrical current. So when we look at pH, the pH scale is shorthand for the hydrogen ion concentration. The scale runs from 0 to 14. Most people are familiar with the pH scale in terms of that. Anything below 7 is an acid, 7 is neutral. Anything that is above 7 is an alkali or a base. You can use either term, but it tells us what it is. So basically that pH scale is the amount of concentration of hydrogen ions. All right, so if you have an acid, you have a greater concentration of hydrogen. The more concentration of hydrogen, that lower that pH, that stronger acid. An exact opposite with base. All right, so now let's go into, now we kind of got the basics of chemistry. Let's kind of look at what our cells are made up of. And we're made up of organic molecules and inorganic molecules. In chemistry, the term organic does not mean it has been raised on some farm that doesn't have, all kinds of chemicals. Organic to a chemist means that it has carbon and hydrogen. That's what we mean by an organic molecule. So when you study organic chemistry, that's what you're studying, is those compounds that have carbon and hydrogen. So, and we'll look at those. So those organic molecules that we're going to work with are going to be carbohydrates, proteins, lipids, nucleic acids. We need those. And they're going to make up, they are the building blocks of our body. We also have inorganic molecules. These are, again, the ones that don't have carbon and hydrogen, and they usually dissolve in water, and those are things like our inorganic salts, our minerals, water, oxygen, carbon dioxide. Those are not considered to be organic molecules. So we're going to go through these organic molecules and take a look at just the basic ones. This is a short little introduction to them. Go through your notes, learn a little bit more about them, but I want to give you a little bit of introduction to these. First of all, carbohydrates. This is our main source of energy. The body is going to use carbohydrates first, and it's going to use up its carbohydrate stores. And when it runs out of carbohydrate stores, it will move to other stores such as lipids and then finally into proteins. But if we have carbohydrates available, the body is going to use these. And this is what is the cell uses for its energy. It's going to be, there's several classifications that we'll look at in a second. But this, again, is the main source of energy. This is how we break glucose up. We're going to look at this in another chapter, another unit, how we actually take that glucose, how we take that carbohydrate and break it up and make energy from it. Okay? So the formula for the carbohydrates is a 2-to-1 ratio, basically of carbon and oxygen should be the same, and the carbon and the oxygen are half of the hydrogen. So you're looking at a formula, you have a carbon, you have a hydrogen, a carbon and an oxygen, and they will be the same number. Look at our glucose, C6O6, and then the hydrogen is 2 times that number. So C6H12O6, that's glucose. C8H16O8, that's going to be a sugar. So anytime you see that ratio, that's going to be a carbohydrate. Now we have different kinds of carbohydrates. First of all, our basic unit of a carbohydrate is a saccharide. A monosaccharide is a single or simple sugar. It is 1 unit. We'll take a look at it in a second. And examples would be glucose and fructose, fruit sugars. Disaccharides are double sugars. They have 2 of the monosaccharide units. Sucrose, that is table sugar. The table sugar that you use on your table every day, that's sucrose. Lactose, that's the sugar that's in milk. Then if you have more, you call them polysaccharides or many saccharides. And these are the complex carbohydrates that you hear about. Starch. If you're an animal, you store your carbohydrates as glycogen. If you're a plant, you store it as cellulose, and it turns out to be kind of like the fiber that you're eating. So here's our carbohydrate. Here's our glucose. There's our straight chain of carbon, 6 carbons. Then we have the 6 oxygens, and we have the 12 hydrogens. Now, you take organic chemistry, we're going to talk about more commonly that the glucose forms what we call a ring structure. We call this a carbon ring, and this is the symbol that shows us that that's what that is. So monosaccharide, you have 1 of these structures. A disaccharide, you have 2 of them with a bond in between. And a polysaccharide, you have more than 2. You've got several. All right, lipids. Lipids are fats and oils. They're insoluble in water, but if you add some kind of solvent to it, they are soluble. They include what we call triglycerides, phospholipids, steroids, hormones. Your steroid hormones are all made up of these. Phospholipids we'll talk about when we get to the cell, because that is a lipid that makes up the membrane of the cell. So these are important components and have a lot of functions in the cells itself. The most abundant lipid is the triglyceride, and that is used for energy. It has more energy per gram than carbohydrates. This is why when you eat fats, you can gain more weight, because it has more calories. And you'll take a look at this in AP2. It does have carbon, hydrogen, oxygen, but it has less oxygen. When we look at a triglyceride, we have one molecule of glycerol and what we call 3 tails of fatty acids, and we'll take a look at that in a second. All right, and you've heard, I know, in ads and that sort of thing about saturated fats and unsaturated fats. What do you mean by that? And I'm going to show you exactly what it is. But saturated fats only have single carbon-to-carbon bonds. So the carbon is bonded together with what we call a single bond. These are solid at room temperature. This is how you can tell a saturated fat. Most of them are animal origin, sorry. But we're talking about lard and butter and that sort of thing. This is a saturated fat. When you move into unsaturated fatty acids, you're talking about more than one carbon-carbon double bond. These are liquid. These are the oils. So these are usually plant origin, like your olive oil and your corn oil and your canola oil and so forth. So let's see what they're talking about. What they're talking about with this is the saturated fatty acids are all lined up in that little line, and there is one bond between each carbon, okay? Which means that the two hydrogens are bonded. When we go into the unsaturated fatty acids, you can see right here, this is a double bond. This doesn't have hydrogens here, and we don't have hydrogen here. So we're bonding, we're filling the carbon's outer shell by bonding the carbon together. All right. So again, saturated fatty acids, solid at room temperature. Unsaturated fatty acids, liquid at room temperature. This is a triglyceride. You've heard a lot about this, but this is what it means. It means this is a glycerol, and then you can see the fatty acids. There's 3 fatty acids tails down there, just to show you what they look like. So lipids, we talked about phospholipids just to introduce this. They are similar to triglycerides. They have a little bit different structure, but as I mentioned, they are a main component of cell membrane. So they're going to be very, very important. They do have a phosphate group on them. That's what makes it different. They have a glycerol. They have 2 fatty acids instead of 3, but we have a phosphate group bonded on. So tacked on. So phospholipid, that tells you there's phosphate group. Steroids, these are 4 rings of carbon. We talked about the carbon ring. This is a component of cell membranes. This is the main steroid in the body, cholesterol, and then cholesterol, this structure is used to synthesize adrenal hormones and sex hormones. So anytime you see testosterone or estrogen or something like that, that's a steroid. That's going to have that composition. All right, proteins. These are structural material. They can be an energy source. They make up some proteins. You'll learn, hormones. You'll learn that in AP2. They act as receptors. All our enzymes are proteins. Our antibodies are proteins. These are important, okay? They are made up of what we call amino acids, and we'll take a look at how amino acids, these proteins are actually made from amino acids in a further chapter. Amino group on one end, NH2, and a carboxyl group on the other end, and then we have what's called a side chain, an R group. And that's what makes these amino acids different. They are bonded to each other by peptide bonds, but you can take a look at this. So here's our amino acid. This is the structure of an amino acid. You have this structure, you have these carbons, you have this carboxyl group on one side, or hydroxyl group, and you have this amine group on the other side of the central carbon. And then this piece right here is what we call the R group. And it is the R group that changes the name of the amino acid. So if you look at this, cysteine is the same structure here, but the R group is different. Phenylalanine, main part is the same, the structure of the R group is different, and this is the same for all amino acids. Okay, so you can see that. All right, so when we make proteins, there's a certain structure to them. We're going to learn how they're actually made in another chapter, but there are 4 levels of structure. First is the primary structure, and that's just the sequence of amino acids. We just looked at 2 of them. We looked at cysteine and we looked at phenylalanine. So we have just this sequence of amino acids. Secondary structure is where they actually pleat or twist. There's hydrogen bonds forming, and the structure either is what we call pleated or twisted. And then as it pleats or twists, it forms this 3-dimensional shape, and we call that the tertiary structure. And then we may get quaternary structure. This is formed by some proteins when you have several of these chains connected to become one protein itself. And I'll show that to you in a second. Now, because of this structure, it's very easy to denature a protein. This is when we change that structure of that protein. We can heat it up. We can use radiation. We can change the pH. Sometimes it's reversible, sometimes it's not. So a good example would be the difference of, for example, milk and a white of an egg. Both of those are made up of proteins. Your milk protein, when you heat it up, milk gets nice and warm, nice cozy drink at night, if you know. And you can also cool that right back down. It doesn't change. You put it back in the refrigerator. There's no change to the structure of that milk. So that is reversible. The white of the egg, you take that white of that egg, you put it in your frying pan, that protein structure is broken apart, and you get the white of egg, and there is no way you can put that egg back into the white of that egg back into that egg. It is permanently changed, so that change is irreversible, and we have denatured that protein. So when we talk about enzymes, which are proteins, if you have denaturization, that protein can no longer function, and we'll talk about that. But here is our primary structure. Here's that secondary structure. There's your twist, there's your pleats, and they all fold together, and this is your tertiary structure, and then you finally get several together and you get a quaternary structure. All right, nucleic acids. We are talking about DNA and RNA, and most of you have been introduced to this, but DNA carries the genetic code, and we'll talk about this transcription into RNA, and then translation, which finally makes the protein. So the nucleic acids encode these amino acid sequences. They are made up, the nucleic acids are made up of building blocks called nucleotides, which are consisted of a sugar, a phosphate group, and an organic base. So first of all, we have DNA and RNA. Here's one difference immediately with the DNA and RNA is the sugar. So RNA is ribose, DNA is deoxyribose. So there is one oxygen left, and that is the real difference when we look at that. DNA is a double chain of nucleotides, RNA is a single chain. So here's our lovely little RNA, there's our phosphate group, there's our sugar, and these are our nitrogenous bases. And same thing with DNA, all right, except DNA is a double strand. The A, most of you know, adenine, thymine, the T, the C, cytosine, guanine, the G, and U, uracil. Adenine always bonds to thymine, okay, in DNA. The thymine bonds to adenine, cytosine to guanine, guanine to cytosine. That's the way it always bonds. Now, when you're talking about RNA, cytosine bonds to guanine. Well, we don't have any thymine, so adenine bonds to uracil. Uracil bonds to A. So we replace this thymine, and that's really the difference. So when we take a look at our nucleic acids, the DNA is the genetic code, whereas RNA is going to help us in protein synthesis. And we're going to talk about and go through that procedure in not the next chapter, but the chapter after that, chapter 4, about how this actually occurs. Okay? So thanks for listening, and we will do the parts of the cell next.